Hi friends, you're back with me, Professor Girish Kukreja. Yeah. So today we'll be talking about one serious problem. Nothing is serious that much in life as we think it is, because maybe we don't know the insights into it, and therefore they seem to be serious. Once we know that how things are working and what exactly is to be done, probably things are very simple. One such thing which happens with most of the microbiologists when they are working with mycelial organisms, and they are preparing the inoculum for these mycelial organisms, and they want a good growth of these mycelial organisms in the submerged fermentation. We confront with the problem that sometimes you have a pellet type of a growth, right, where you have some discrete masses, a compact mass of these mycelial organisms which are growing, and otherwise the broth is what you call as somewhat more clear, right? But then you have a more homogeneous type of a growth, where which we will refer to as the filamentous type of a growth, right? So where you have a more dispersed growth, we can say. So here you have a pellet shaped growth or a pellet type of a growth sometimes, where you have a compact mass. Otherwise you have a filamentous type of a growth. Many of the times when we want a pellet, we get a filament. When we want a filament, we get a pellet. This is what happens. We never get what we want in our life, and then we think that we should be happy with what we have. Yes, that is also true sometimes, but then you should remember that we can also try and find out that how exactly we can get what we want, because probably we don't know the way. So here also, as I told, then what is right, whether it is pellet or whether it is a filamentous, right? Which is a better option for me, right? Again, it depends from organism to organism. Each of these forms will have their own advantages and disadvantages. Like if you go with the filamentous form, you will get a more homogeneous kind of a broth, where you'll find that the broth but then becomes more viscous. It becomes more problematic for aeration and agitation. With pelleted form, probably you'll find that the broth is more clear, but then now it is not homogeneous. So you get one and you lose the other. And but then you'll find with the pelleted, another problem is that you'll find that the cells which are there inside that entire compact mass, they may die because of starvation, because of oxygen requirement, because probably there are diffusion problems at the center. So what is better, as I told? As I told, that one thing does not work for all. You know. So say for example, like if I am going for production of penicillin by Penicillium chrysogenum, it is expected that this filamentous form which is there, that is more appreciable. This gives me a good yield. Reports have said that the pellet form is more better when I am producing citric acid from Aspergillus niger. So maybe once I know that which form is better now for my fermentation, like if I am going for penicillin production, I need a filamentous form. If I am going for Aspergillus production, I'll go for the pellet form. So now the question is like, how do I control, right? So that is a major thing. We want everything in our control, but have we really known the mechanisms that how it is functioning? Okay, so probably there are two things which mainly affect this. One is the spore size, right? The amount of the spore which you are inoculating, and another is your medium. Uh, Again, there are no thumb rules in biology, right? In biology, we are very flexible. There are no thumb rules as per the situation, as per the environment, as per the organism. Even with the same organism, the things keep on changing. So, no generalization as such. But then, yes, there are some things like which are more what you call as applicable at different places. So, as far as spore is concerned, one a very general, a very vague kind of a thing you can say is that the more you have the spore inoculum, the higher is the spore suspension. You have a more probability of getting a filamentous kind of a growth, right? And you have a lesser what you call as quantity of spores. Probably this will result in a pelleted form of a growth. So I have one control in my hand now. So a higher spore, a filamentous form, a lesser spore, a pelleted form. Mostly, again, I'm telling, there are many other things which will come into picture. The shear force is another and all that stuff. I'm talking about these two right now. Medium, as far as medium is concerned, a rich and a complex medium, it will result in a form of a dispersed growth, what we refer to as the filamentous growth. and a chemically defined, a simple medium, a chemically defined medium, it will result in a pelleted growth. Now I have two keys. So as I told that I can control this to some extent. Again, as I told that this you need to verify at your end also, because this may vary, as I told, with the conditions of the organism. But in general, let's remember that if I want a filamentous or a dispersed growth in the medium, I will use a higher spore inoculum, and if I want a pelleted growth in the medium, I would use a lesser spore concentration. If I want a dispersed growth, I'll use a rich and a complex medium. And if I want a pelleted growth, a simple and a very defined chemical medium. So maybe with this, I hope that it would be now a bit easier. Let's try this in our laboratories and see whether this works for me or not. Right? Stay tuned with me, Professor Girish Kukreja, for more in microbiology and biotechnology. Thank you.